Okay. We made it on this meeting. So we're gonna talk about plants that'll warm us up. Today in lab, we'll learn how to dye fabric, wood, using plant material to dye. And then Thursday, you'll be doing propagation. So this slide is relevant to that because the goal on Thursday, most of you will plant, and then we're going to put those stem cuttings in the water so that we can witness the adventitious roots starting to form at the base of the stem. And so as we were learning last week, we started looking at plant growth. And so we introduced the idea of Darwin, working with how plants respond to light. Once they discovered that auxin tip. They didn't name it auxin, that came later, but they were credited first significant real research on looking at this phenomenon of how plants will bend toward unidirectional light. So when you take a stem cut, it's really quite remarkable. You're going to be placing the stem, inviting to give it a little dose of rooting hormone, and then you're going to put it into a little jar of water. And what's happening is it punches up as that cut base of the stem. So at the base of the stem, those are cells that were stem cells. Well, now they have to, the fancy word is called de-differentiate. But the fancy word is de-differentiate. Those cells that were designed to be stem cells now have to switch root cells. So we have to de-differentiate being stem cells and then kick in all the necessary gene expression to become roots and start growing out these adventitious. So it's really quite hard. Fingers, we can't. And here it's important because stem tissue that stem, not root, and yet it will totally switch and become root tissue. So it's really quite remarkable. And auxin does help with that. So if you decide to dip that cut stem, we didn't call it that powder is mostly auxin hormone. That is going to develop this habit, but it just kicks that into high. And so here you're seeing a few different images of adventitious roots. So when we say adventitious, it means they weren't destined to be root cells like in the seed. You know, we talk about the radical embryonic roots. They're not in the tissue. They were stem tissue. That's now root tissue. So we call them adventitious roots to separate them from the idea of they weren't initially root tissue in the embryo of the seed. So here you can see orchids are famous for having some adventitious roots. Often their roots will actually start to turn green because a lot of orchids that, most orchids actually, they are found in tropical regions and they live on the branches of trees. And they're not harming those trees, they're just using those trees to get further up off the ground to get access to light. So their roots are just dangling, they're not in soil. And you'll often see orchid roots are green because they're actually doing photosynthesis. So that's kind of bizarre. And here you can see in the top left, that's a tissue culture vessel that that the plant tissue is in. It's starting to form adventitious roots. And then here you can see a vining plant. The stem tissue is on the lower left, starting to form those adventitious roots. And in this case, it's for if they latch on to like the side of a wall or another tree, they can use those adventitious roots to grab on and stabilize a little bit. So auxins are very famous for adventitious root formation, but they're also really heavily used if you want to clone a plant through tissue culture procedures. And so that's what you're seeing in this image. It is a test tube that is full of a agar media that supports, it has all the ingredients to support plant growth and cell division, including auxin hormone. And so when I teach plant physiology, we do a plant tissue culture exercise where students will clone, tried a variety of different plants. Lately, it's been African violets and tomatoes. And you have to provide the right recipe of ingredients and the right ratios and types of hormones in order to get the plant cells to divide. So what you're seeing here in this white clump, that is what's called a callus. And a callus, when you say that in the plant world, that just means that it is plant cells that have grown and formed a mass, but they haven't differentiated into the different parts they're destined to be. And so it kind of just looks like a blob of cells. And then with the right ratio of hormones, you can then tell those cells, okay, start differentiating. Some of you become roots, some become stems, some become leaves. And so auxins are heavily used in the plant tissue culture mechanism because their auxin hormones are very good at stimulating cell growth. Divide, grow. So that hormone is very good at that. They're also involved in phototropism and gravitropism, which I'll discuss in the slide set. We have that coming up soon. But phototropism, tropism of movement, that's kind of alluding to this idea of movement. So phototropism, moving toward unidirectional light. Gravitropism, the idea of the plant sensing and responding to gravity appropriately. When I say that, I mean the roots have to grow down toward gravity, but the shoot has to grow away from gravity. So we'll talk about those ideas. So let me give you a survey of some of the other hormones. So gibberellins often is referred to as gibberellic acid. Gibberellic acid. And so that's why we often abbreviate it GA, right? So they're involved, their claim to fame is stem growth. And so here you're looking at an image of cabbage, and there are some varieties of cabbage that are called dwarf cabbage. And so normally they'll just grow in a dwarf configuration. The whole plant will just stay kind of short. And then if you take some of those dwarf cabbages and spray them with some gibberellic acid, it causes the stem to grow tremendously. And so that was one of the first things that they noticed, and then they discovered it was gibberellic acid doing that. They actually discovered this in a rice paddy when they were growing rice of their conventional systems, rice paddies. They noticed that some rice plants got way too tall very quickly, and then they flopped over. Well, if you go to harvest rice and the rice plants are all flopped over, it's really hard to harvest. So disturbing, aggravating thing. So they're like, why are these plants doing this? And so they went and started looking at those plants, realizing that a fungus that was affecting the plant, and the fungus produced gibberellic acid, which was causing the plant to grow too fast, too quickly in the stem, and then that caused it. So it was actually first discovered in a fungus that hangs out with plants and causes problems. And then they realized that the plant itself also has this hormone. That's why it was able to respond to the hormone coming from the fungus, because it has all the necessary mechanisms to sense and respond to this hormone. So that's how they first discovered gibberellic acid. And there's many different types of gibberellins out there. There's not like just one type, just like with auxins. There's lots of different auxin hormones that fit into the auxin category. So when you look at, when we do this in plant phys, we don't work with cabbage. We tend to work with pea plants. They're smaller and don't take up as much space. And so here you're looking at dwarf pea plants on the right. And then if you spray those dwarf pea plants with gibberellic acid, they shoot up in stem length. And it's really noticeable. You can't help but see the difference and measure huge increases in stem growth. So it is responsible for stems. And of course, you need good stem growth if you're a plant, because you need to be able to get access to the sun. And then what if somebody's shading you? You might want to grow up a little bit more. And so this hormone is really important for the plant. Here, they've taken advantage of it in the commercial grape industry, because if you spray the grapevine, which is stem tissue, with gibberellic acid, it causes the stem especially the area, the stem tissue where the grape clusters are starting to form, where the grapes are forming. It causes that region of the stem to space out more, so it gives more physical room for the grapes themselves to get bigger and typically sweeter, which of course gets you more money when it goes off to market. And so that's a good strategy of how they use it.
So, so make it so that, I know I was getting my
Yes, yeah. So these days, most people want to see the grapes, right? And in fact, when you get to the end of the semester, these are grapes that I can't find, hard to find. So we have kind of given up on that idea. But and right, I mean, I like seedless grapes. They're really easy to eat. But you can see the difference. The one on the left was not sprayed, and the one on the right, that vine was sprayed. So that tissue spaced out enough. The grapes grew bigger. Are you talking about impact on our health, like when you eat? So it's kind of interesting because as far as digesting it, I have not read any negative impacts. And in fact, I've seen actually they're starting to use plant hormones for skincare, like skin lotion. And if you go online or sometimes I can see it in stores around here, I have fun wandering the skincare aisle. As I'll often talk about adding chitin, which is a cytokinin hormone. And they are looking at, there is some research looking at some plant hormones in human health in a beneficial way. I can post some or send it via email for anyone who's interested. It's not mandatory reading, but if you're curious. So they claim that putting like cytokinin type hormones, which we're talking about, into skincare and helps rejuvenate the skin cells, make the wrinkles go away. I don't know. There is some evidence, but I would like to see some really good controlled scientific studies on that, rather than a company just claiming it to be so and then charging triple the price of what it really costs. So I'm a little skeptical, but I'll send you some papers that talk about this idea because I've wondered about that too. What does it do to us? And so far, I've been finding good things. Gibberellins, hopefully your seeds are germinating. If you haven't already planted them, engaged, already germinating when you go in there today. Make sure you water your plants or your seeds anyways. If you haven't sprouted, keep watering that soil. You got to keep it evenly moist. And here you can see this beautiful series of images looking at how when the seed first germinates, it's the radicle or the embryonic root that comes out first, which makes sense if you think about it, because this seed has, usually most seeds have nutrition packed into them that they can rely on, but they don't have water. I mean, if they had water in their seed, then fungi would attack them and kill off most of the seed. So they desiccate in the process of seed formation. They want to desiccate, dry out, so that they're less of a target for fungi to attack them. And so they're desperate for water, so to speak. And so it makes sense that the radicle comes out and starts searching for water immediately. That's the number one priority. And you can see how they form really good root hairs, because that's the part of the root that really does a good job of taking up water from the surrounding soil. And then you see the baby seedling starting to sprout. So they also know that as the seedling continues to grow, the gibberellic acid present in the plant will help regulate the shift from what we call the juvenile phase into adult phase. So in the animal world or human world, we tend to talk about puberty versus adulthood, right? It's kind of the analogous idea. Juvenile phase means that they can't yet go through sexual reproduction. They don't have that ability yet. And then as they shift into the adult phase, then they can do that. Okay, so gibberellins are known for regulating that process. They're also involved in cell division. So similar to auxins, how auxins help cells divide and grow, gibberellins play a role in that as well. And so here, if you've taken BI-111, you've learned about mitosis. And here you're seeing under the microscope some cells that are going through mitosis. So that's the way that cells grow and divide. It goes through mitosis and produce two progeny cells from one cell. So, cytokinins, I'm sorry. Cytokinins get into that idea. So with gibberellins, I think I have some slides further on where I show some other impacts that they have with how we use them in the commercial industry. But let's get into cytokinins with cell division. So when you look at tissue culture recipes, if you're going to clone a plant through plant tissue culture, typically what they have in the recipe is auxins and cytokinins. And it depends on the plant, how much of each, and what ratio. And then you can pick recipes that just have auxins to get things going, and then you can switch the tissue onto a different recipe that has cytokinins. So when I teach, often I like to do one hormone at a time, so you can see the effects of this hormone. Okay, then what happens if you add this hormone? So for teaching purposes, kind of fun to see that shift. And when you see the auxin effect, it's usually just cell division, and they form that callus. And then often, if you take that tissue and put it on tissue culture that's cytokinin in it, that continues cell division, but then it starts to differentiate and grow into stems, roots, leaves. And so there's fairly high quantities that are produced in root tips of plants. That seems to be a popular place to have the cytokinin being produced. But then you can also find it in other parts of the plant. And so young leaves, developing fruit, embryos. So again, with hormone research in plants, it can get quite complicated because you can find one hormone in many different parts of the plant body, being produced in many different parts. And that's kind of not so much like what we see in animal hormones. They tend to be located in one region. They're famous for their ability to stimulate the axillary buds or the lateral buds to grow. And so auxins, we learned how auxins are keeping the axillary buds dormant because auxin in the shoot tip displays apical dominance. And so what happens is auxin is flowing down the stem and all the axillary buds are quiet. Now, if the right conditions arise, and it's usually a multiple things the plant is sensing, including its environment, temperature, light, duration, all of that, then you can get the cytokinin upregulating in the axillary buds. And if the ratio of auxin to cytokinin tips the scale in the right direction for the axillary buds, then the axillary buds will start to grow. And so again, it's when hormones are involved, you have to appreciate that the plant is constantly assessing its environment, and it's exquisitely sensitive to its environment. And so it has lots of ways to figure out what the day length is, night versus day, temperature, if it's getting shaded or not, what's the soil water like. All of that is going on minute by minute by minute. They are way more sensitive to the environment than we are. And we've kind of lost that ability because we don't really need to pay too much attention to it. We have the ability to be mobile and get to somewhere safe if the conditions become threatening. Plants don't have that. Chloroplasts, of course, are very, very important for the plant, kind of a key thing for the plant to have up and running and fully healthy. And they know that cytokinins are important in getting the proper development of chloroplasts. Cytokinins in plant tissue basically are telling the rest of the plant that tissue is healthy. So in spring, when the leaves first bud, or they start out, and then all summer long, the leaf tissue has a fair dose of cytokinin in it because it's important for the leaf development, the chloroplast, all of that. Now in the fall, when we see the beautiful fall colors here, what's happening is the cytokinin levels are dropping in the leaves. So if cytokinin is there, we see it delays senescence. Senescence is the natural death of parts of the plant, cells. So it's natural with our deciduous trees that in the fall they go through the process of siphoning out all of the nutrients in the leaves and then dropping so they can go dormant in winter. So that's leaf senescence. Well, so in the summer, there's plenty of cytokine in there basically saying, hey, we're healthy, we're good, don't get rid of these leaves yet. But then as cytokinin levels drop, that's when you see the shift in color as the leaves are going through the senescence process and falling off eventually. Here on the right image, you're seeing an experiment where they have the mechanism where there's a drop in cytokinin on the plant on the right, and so it aged and senesced much quicker. So that's why this aging idea is another reason why skincare companies are getting into this idea. Maybe cytokinins would delay the aging process in animal cells as well. Another one to consider is this one. Now, this is just one hormone. It's not a group of hormones like what we've been seeing so far. Cytokinins, there's a lot of different cytokinin varieties of hormones that fit into the cytokinin group. Abscisic acid is just abscisic acid. It's ABA is how they often abbreviate it. I like to think of this hormone as a brake, whereas many of the other hormones are like a gas pedal. This is more like a brake pedal, where it is responsible for like seed dormancy. So when you took your seeds and you got them out of the package, right then the abscisic acid levels are fairly high. But as soon as you wet those seeds, and certainly now if you planted those seeds, the abscisic acid level is dropping tremendously in that seed. It also can regulate other hormones by kind of slowing things down, inhibiting, putting the brakes on when it interacts with some other hormones and what they're doing in the plant. So acts like a brake is a good way to think about it, like a brake on a car or a motorcycle. So seeds in general have abscisic acid present when they're not ready to germinate. It's kind of like telling the seed, stay quiet, it's not time yet. When you wet those seeds, when you put them into the paper towel, the wet paper towel, what you started doing, that all that moisture started to kind of dilute out the abscisic acid. And so abscisic acid levels started to drop. And so under normal conditions, those seeds will stay dormant with high levels of abscisic acid. Same with the buds on the trees. Like right now, all winter long, the buds have a fair bit of abscisic acid, just telling the buds it's not time yet. It's not spring. Don't go ahead and germinate and grow. Another very famous effect that abscisic acid has is that it regulates the guard cell movement. It helps with that. So that's what you're seeing here. You're looking at epidermis tissue, and you can see those lovely kidney bean shaped guard cells surrounding the pore, guarding the pore. These other cells right around the guard cells, those are called subsidiary cells. You don't really need to know that, but sometimes some plants will have extra cells right there to kind of help. So what's happening is abscisic acid is actually synthesized in the roots. It can be found in root tissue. So imagine that you're a plant, a typical plant that has your roots, you're in the soil, your roots are in the soil, and it's, let's say it's June, and your roots are starting to pick up on the idea that, hey, there's not much soil or water in the soil. We're kind of getting into the drought phase. What you do then, your roots will start kicking in and making abscisic acid in response to that drought. And then very, very quickly, you'll send that abscisic acid up to your leaves and you'll say, hey, guard cell kind of pose. Right? So that kind of makes sense, right? If you are under water stress, you can't get enough water, you don't want water leaving your body. That would not be smart. And so if you're under drought conditions, close down your stomata. So that water can exit out through the pore. And that's exactly what they see happening. And it's the abscisic acid is made, synthesized in the root in response. And so it happens very quickly, and then it has to course through the vascular tissue and get up to the leaves and tell those stomata, hey, close down, we're under drought. So it's a very effective mechanism to make sure that you don't have too much water through the plant and out into the atmosphere. So we'll talk about water movement more as we get into roots and stem tissue. Let's start thinking about that. Okay. So the last of the classic hormones, usually we teach in an intro botany course, we call them the classic five. These were the first five to be discovered. The last of the five is ethylene. Of the classic five hormones that we've talked about to date, ethylene is the only one that's gaseous. It's a gas. And it is produced by the fruit, tissue, flowers, seeds, leaves, and roots. So you can find it being synthesized in multiple parts of the plant body. And it can kind of do its own feedback loop. So as ethylene gets produced, it kind of feeds back into the pathway and kind of ramps up more ethylene, more ethylene. Ethylene is famous for ripening up fruit. It's also famous for the idea of getting leaves to drop off of the plant. So that process is called leaf abscission. Abscission means to, you're going to see, I think there's a picture of an abscission zone in the leaf petiole that weakens and causes the whole leaf to fall off. So leaf abscission is just falling off of the leaves. It's a natural process during senescence. That's part of the process. So here you're seeing holly leaves under a beaker. And so what they did on the left one is they just have holly leaves under a beaker as the control. And then the right is they put an apple, and they probably cut it or bruised it, somehow damaged the apple, and then put it under the glass beaker with another holly plant, and you're seeing the leaves falling off. So what's interesting is that if you cut or damage an apple, it immediately starts to synthesize ethylene in response to that. And so ethylene is coming off the apple. If you enclose it with glass, it'll stay right around the holly plant, and really, in this case, it's promoting leaf. So you can take advantage of this in your own home. To actually fall off, I'd say maybe a week. I do this at home a lot. Like I just went to the store last night and got the only option were green bananas. Have you noticed that? When you're at the store, sometimes it's like, oh, green, that's your choice. It's like, okay, so what you can do if you want a more ripe apple quickly, take an apple at your house, cut it. So when you cut it, you're wounding it. Ethylene is produced in response to wounding, whether it's a wound from a human or an animal chewing on it, whatever. Put it in with your cluster of bananas and somehow enclose it. Paper bag is probably better than plastic, so there's less humidity buildup. But that's enough to trap the ethylene around the banana, and you can get ripening up. So if you're impatient for more ripe bananas, give this a shot. And it usually does respond. Bananas are really sensitive to ethylene, and they will respond pretty quickly. So ethylene is a gas, so you have to keep that in consideration. You want the gas to be around whatever fruit you're looking at, you have to enclose it somehow. By the way, some fruits don't respond to extra ethylene. Some do and some don't. Bananas are really good at responding. Others seem to be a little bit more immune to it. So ethylene is produced in fairly high quantities with any kind of wounding or stress event that the plant is experiencing. It could be wounding, it could be flooding. So too much of anything can be bad, including water. Ethylene can also get synthesized if the right amount of auxin and cytokinins are in the tissue. That tends to stimulate ethylene. Here you're seeing cut flowers and how ethylene actually promotes leaf senescence. So we talked about how cytokinins delay it. There's enough cytokinin in the leaf of flower, it keeps that leaf or flower healthy. Once cytokinin levels drop, ethylene comes up, and then you'll see senescence. So in the cut flower industry, if you're trying to sell cut flowers for your living, you do not want ethylene in your business building. And like you see, flowers are in coolers. You don't want those coolers filled with ethylene. And so people that run businesses in the cut flower industry, they have ways of trying to minimize ethylene impacts. One of the things that they do is in this beaker of water that these cut flowers are sitting in, you can put this powdery mixture that will kind of sop up the ethylene and keep it from impacting the cut flowers. So if you're in the cut flower industry, you better do your research and know what can you do to minimize ethylene around your plants, your cut flowers. Because certainly you don't want to sell flowers that are going to quickly die. You're not going to have a very good business reputation if you sell flowers that are on their last legs, so to speak. So ethylene is very famous for that. And so in leaf abscission, when we talked about that with cytokinins, how it delays senescence, what's happening is when we see the fall colors, auxins and cytokinins in the leaf tissue start to go down. And that actually, ethylene synthesis starts to go up. And then it's actually ethylene which is telling that leaf, okay, it's time to fall off the plant. Go ahead and go through leaf abscission. So it's a whole mixture of different hormones impacting the leaf tissue in the fall during the beautiful fall colors. Okay, so those are the classic 5 hormones. There are other. Haven't taken vitamins, you'll learn more about them. They are discovering other hormones that be incredibly important both for the plant and the industrial industry of how to work with plants. So, for example, jasmonic acid, familiar. There's others, strigolactones. There's a handful of others that have been discovered, and some that have been discovered that are really, really important for the plant to fight off pathogens or pests that are a pathogen. And so some hormones are involved in defense, trying to protect themselves. And some hormones actually can be used to warn nearby plants that they're going to be attacked next. So if a plant is getting attacked, let's say a caterpillar is chewing on a seed, the plant can actually determine it's a caterpillar rather than a bunny rabbit or an ant or whatever. It can tell because the caterpillar is oozing out chemicals in its saliva as it's chewing on the plant. And the plant is that sensitive where it picks up that signal and go, oh, I'm getting chewed on by a caterpillar. That kicks in a whole cascade of hormones and other secondary metabolites to defend itself. Some of those hormones get volatilized, go off into the environment. There's nearby plants that can understand that signal. Those plants start to ramp up their defenses against that exact same method. So it's a way for these plants to communicate with their neighbors that, hey, I'm getting attacked. You're probably next. Ramp up. And when this first was published, the scientists first published it, everyone's like, no, this is not. You must have made a mistake. You're wrong. And then they did a really careful controlled study in the lab where they set up controls and then for experimental treatments. The greenhouse-based study was able to support what they call it when they were studying. So now it's more widely accepted and there's good data to support this. It's really irrefutable that these plants are capable of volatilizing some hormones and chemicals to communicate with others around. So I just think that's really, really cool. So plants are rooted, of course, the typical plant has their roots in the soil, but they can still move. So this is a book that was actually published in 1880 by Charles Darwin and his son Francis, The Power of Movement in Plants. So they're really credited with kicking off this field of study. How do plants move in response to certain stimuli that they're getting from their environment? So they were the ones who were first like, why is my plant bending toward the window of my living room? And so they were like, what's up with that? What's causing that? And so they started studying that phenomenon and they wrote a whole book about it. So that begs the question, well, how do you move if you don't have muscles? I mean, we have muscles, so it makes sense, that we have really good movement, but plants don't have muscles. So what are they doing to allow for this movement? Basically, there's a couple of things. Trigger pressure, water pressure. So when you have a well-watered plant, you have the tissue well hydrated. Do you remember what organelle plant cell is packed full of water and is really important in a mature cell? Sure. The vacuum. So in a mature cell, you have a vacuum. If it's well watered, then forms of water pushing against everything else in the cell, pushing everything else off to the side, against the cell membrane, against the cell wall. So you have that water pressure. So we say turgor pressure. It's a fancy way to say it. So turgor movements just mean that some parts of the tissue will lose turgor pressure, other parts will gain turgor pressure. So you'll get this shift and it can cause a movement in that part of the plant. So turgor movements, I'll show you some examples. So it's a shift or a change in the internal water pressure when you look at that part of the plant body. So it's often this asymmetrical shift. I'll explain that a little bit more, but basically you want loss of turgor in one part of that tissue and gain of turgor in the other part, and then that will cause an asymmetric turgor pressure, which allows for movement to occur. So I guess let's talk about it right now. This right image here is looking at the a petiole here. And what you're seeing is some parts of that little, a petiole is like the stem to a leaf. It's the part that the leaf blade attaches to the stem. We call it a petiole. And so the petiole will have this idea of some parts of the petiole cells will start to lose turgor pressure. And then the other side gains turgor pressure. And that will cause a movement of that tissue, an asymmetrical bending movement in response. Here you're seeing on the bottom images, guard cells and the pore, so the stomata. So when you think about guard cells and how they move, they are also responding to turgor pressure. So what we see and it actually, when I first thought about how guard cells move when I was a student, I had it totally backwards. What happens is guard cells have these special built up strands of thickenings, microfibrils, in their cell wall, and it's a radial pattern. And so because of the way those microfibrils are laid down in the cell wall, what happens is as water rushes in to the guard cells, so if my hands are guard cells and right now they're relaxed and there's no opening, the pore is totally closed. If water rushes in to the guard cells, what it happens is they gain turgor pressure and it actually causes the guard cells to bend away from each other. And then that reveals the pore, okay? So it's kind of weird, right? It's like you have to get water rushing into the guard cells to get them to bend away from each other. And then the pore is open. And then if you're a plant going, I'm going through drought, you want your pore closed, that means you have to cause a situation that causes water to leave the guard cells, and then the guard cells will go flaccid, not much turgor pressure at all, it pulls over the pore. All right, so it's just water rushing in and out of the guard cells. that's causing the guard cells to either reveal the pore or close the pore. Water rushes in, it reveals the pore. Water rushes out, it closes over the pore. And they can do, remember, guard cells can be 100% open, 50% open, 13% open, any gradation in between. So guard cell movement, incredibly important for the plant. They have to have exquisite control on that because they can't get up and go to the refrigerator to get more water. They have to be very, very sensitive to assessing how much water do I have in my body, how much water is in the soil, what should I do now in response to that with my guard cells that are guarding that pore. So the pulvinous tissue is on the right here. They had to give it a special word. It's essentially just cells, parenchyma cells, that are responsible for this loss and gain of turgor on either side of the stem, and that causes that bending movement. So turgor movements, that's one way that you can get plants to move. The other way is just grow. And to our eye, sometimes that movement's pretty slow. You'd have to sit there and look at them, stare at that plant for long stretches of time before you could see it. So you have to appreciate that plants are on a different timeline than we are. They just live their life more slowly in most cases. There's some exceptions to that, and we'll talk about that. But growth movements, they can move through growing. And so the roots and stems, you have, when I say grow, when I say that word, it's cell division, where one cell goes through the process of cell division to produce two cells. So cell division is a way to grow. Also just cell enlargement, just getting that cell a little bit bigger is a way to grow. And so there's really two methods of growth, cell division and then just enlargement of the cell. So we will look at some ideas of how they're moving next and go through some ideas of, oh, well, how are they doing that? Let's put it into context. Like, what do they do when they're faced with unidirectional light? What are they doing when they sense gravity? So let's go through some ideas of how they're moving in response to their environment. So we're going to get into gravitropism and phototropism. So growth movements, when you see these happening in the plant, and they usually do it through, they video the plant and then they speed up the video so that it's easier to really appreciate what the plant is doing. But you get varying growth rates in certain parts of the tissue that will cause perhaps a bending response or a coiling response of the plant. And so we'll work through this, but it's usually, again, some kind of asymmetrical growth. We're like, again, looking at stem tissue, some parts of the stem speed up their cell division, other parts slow down, and that will cause a bending or coiling. Okay, so growth movement is usually an asymmetrical growth response. So one of the first ones that Charles Darwin investigated was this idea, where vining plants, like a pea plant, for example, if you put, here he put a stiff piece of paper with some carbon dusted on it, and then he let this plant grow toward that. And as the tip of the plant made contact with that cardboard that had the carbon on it, the carbon dust, it traced a pattern. So again, very low tech, nothing fancy here. And yet Charles Darwin was able to show that these plants are not just growing straight and that's all they're doing. They're actually moving in a coiling pattern as they're growing, and or circling movement, I guess I should say, not coiling, circling movement as they're growing. And so that they gave it the term nuptation. So that's the circling or spiraling movement that you don't really see. You'd have to like sit there for days to see that probably. So that's why we use videography to speed it up so it's easier to see. So Charles Darwin was the first one to come up with that. So as you're studying these links that I'm providing, if you open up the PowerPoint and look in the notes section, I'm going to be providing links, YouTube links, to look at some of these movements. You'll see it, the speeded up videography. It's really fun to watch. And so another movement that young seedlings often do that you can't necessarily see our eye and sitting there watching it is called nodding movements. And it's usually young seedlings that just germinated. And what they think is going on is that you'll see in the video that the stem is leaning kind of back and forth, back and forth, back and forth as it's growing, moving. And if you think about it for a second, that kind of makes sense, because if you're a seed buried in the soil, you're surrounded by this soil, but you have to get your stem and leaves above the soil. Sometimes that can be challenging depending on how packed down the soil particles are, right? And so if you have this heavy weight of packed soil as you sprout, you're going to want to try to loosen it up, right? And so that's what they think is a strategy for these baby seedlings to emerge from soil. That nodding movement helps break up soil particles so that they have an easier time finally breaking the soil and getting their leaves access to that precious sunlight. So it's really cool. And please watch the video. So that YouTube link should be active, and you'll see there's just short little video clips, and just to prove to you that they have this movement. We can't really see it with our naked eye. You have to speed up the video. Okay, so you're gonna hear about, well, you already have heard about tropisms, phototropism, gravitropism. What are we talking about here? These are movements that the plant is making in response to something. So phototropism means it's movement in response to light. Gravitropism, movement or growth in response to gravity. And so we say permanent, I kind of take issue. That's how they define it, but it's like, well, not really. Because if you do the experiment where if you shine a light on unidirectional light on the plant, it'll start to bend. But then if you take the light and shine it on the opposite end, it'll bend that way too. So it's not permanent per se. I don't know. I would forget about the word permanent. You can actually manipulate that. So tropism, it's growth. It's growth movements, and I'll explain that more. Toward or away from something. Obviously, if you're a leaf and stem, you want to be growing toward sunlight or light. But if you are a leaf or stem, you want to grow away from gravity. You don't want to grow toward gravity if you're a stem. You want to do the exact opposite, because if you grow toward gravity, you're going to end up in the soil. So let's look at this idea, phototropism. So that's the idea that if you have unidirectional light coming through your apartment window, the plant is in the windowsill, you'll have that plant bending toward that light. So that's actually a growth movement. That's a growth movement. And so here you're looking at a plant where the light must be coming from the left or the right. Yeah, it's coming this way from the left, and the plant is bend toward it. So this is actually kicked off. Phototropism is actually kicked off by the auxin hormone that's in the apical meristem tip of the plant. And what it's doing is it's actually causing the dark side of the stem tissue to grow faster than the lit side. All right, so let's take a look at that. So whenever I say the dark side, I always think Darth Vader. So that helps you with this idea. Run with it. I used to watch Star Wars over and over. So anytime I hear the phrase dark side, I can't help but pop into Star Wars. But what's happening is auxin, if you look at this left image, auxin was stained with this blue dye. Auxin, hang on just a sec, we got a minute or two. Auxin flows over to the dark side of the stem, starts flowing down the dark side, stimulating all those cells. Grow, 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 grow. The lit side isn't getting that. You can see the lit side's clear. So what's happening is the lit side slows down and grows, the dark side speeds up, and that causes the bending toward the unidirectional light. And we'll talk about this on Thursday and get into some other growth movements and other tropisms.